So, Mike, I've been interested in things regarding living cells that maybe not that many people, except for you and a bunch of other people, are interested, namely the electrical and electromagnetic design involved in cells and the cell functioning. And much of it was done initially mm, by computational or theoretical means. And more recently, I've been doing experiments um, in my lab and with some collaborators. And I'll try to cover this territory today in this presentation, and stop me at any point if you wish to get deeper into it or after. So, um, this is just the various agencies in Canada and beyond, and companies that were providing support for this work. Um, and more coming up. I think there are two basic aspects, namely to understand, so the basic science behind electrical properties and electromagnetic properties of cells, and different types of cells, as you know very well, have different properties. Um, and the second is applied aspects. How do we harness it for um, A, therapeutic, B, maybe technological, and C, also diagnostic aspects of our health. Um, this is the outline. I don't know if I'll cover everything, but I'll try. So my main interest for the past 2 decades in terms of the structural elements of the cell are microtubules, actin filaments, and motor proteins. So the machinery of the cell in terms of mechanics, morphological changes, and dynamics as well. And microtubules in particular and tubulin have been the central structures I've been focused on. And we did some experiments um, on their electrical properties, electrophoretic mobility. And we discovered in the process, as I'll try to report, that all of this depends really on the ambient conditions. So some of this was done in cells. Sometimes we used purified protein in solution, and the type of solution that was used affected the results. So I think this is one lesson that maybe before we even jump into the data, I would like to convey, namely, in biology, there is really no such thing as the object and the environment. It is interacting always with the environment. So that's main difference in physics. Even in physics, it's not quite true, but we can get away in physics with saying that this is the system and this is the environment. In biology, the system and the environment pretty much is one thing. And I'll try to give you some examples of this later on. And so then we measured conductivity and capacitance, properties that physicists love, and I'm a physicist, so I wanted to understand how that plays out. And then in the second half, I will try to discuss other things such as electromagnetic field. So it's a big difference, and sometimes I really have to spend a little bit of expend effort to explain to non-physics audience the difference between electric fields and electromagnetic fields. It's a major difference. Um, and then in the past few years, we looked at um, um, quantum effects. I don't know if I'll have time to cover that, maybe a little bit towards the end, but there's also the aspect, the enigmatic and highly intriguing and controversial aspect of whether biology and quantum physics have anything to do, anything in common. And I believe that's the answer is in the affirmative, but how much and how so, these are still open questions. And at the end, there are some, I want to talk about some aspects of maybe therapeutic advantages of understanding electromagnetic properties of cells. And the big goal, the major objective of mine, would be to uncover a blueprint for cellular electrodynamics. I know that you've been working on electrical blueprint for especially membranes and ion channels and how that can be understood as an electrical circuit, and that's hugely important. But I think inside the cell there's another electrical system which also interacts with electromagnetic fields. Okay, so first of all, as this slide illustrates, microtubules for me have always played a major role because they in cells because they do play so many different functions. This is a hugely interesting structure with multiple functional roles in cells and living systems in general, and they provide cell shape, rigidity, so mechanical properties, morphological properties, and they are instrumental in dividing cells by forming mitotic spindles, as shown in the middle. Actin is also important, is illustrated in blue in the central panel. And then motor transport so motor proteins such as kinesin, dynein, travel or move processively on microtubules. That means they are moving in a given direction. Kinesin to the plus end and dynein to the minus end. All of this is well known, and great credit to cell biologists to uncover all these facts. But underneath all of this is an expanse of electrical properties, because as you will see here, in addition to being microtubules being structurally interesting, they are formed from alpha beta tubulin heterodimer, and which polymerizes to create these cylindrical structures, typically with 13 protofilaments or these strands, vertical strands in the illustration. Sometimes fewer, sometimes more. It's not always as symmetrical in cells. Things can get messy and you may have defects, and I don't want to get into this territory too much because that's a separate area, but it's important to keep in mind that sometimes these beautiful illustrations need to be taken with a grain of salt. However, what is not to be taken too much with a grain of salt is that they are highly charged electrostatically, microtubules and tubulin in particular. And this graph illustrates the special character of tubulin in terms. Let me just use the pen. Um, oh, I don't know. Oh, doesn't work. I don't know why. But what I want to say is that, okay, number one, the net charge on a tubulin dimer, which is the building block of microtubules, about 8 nanometers long and 5 by 6 nanometers in width and depth. This minus 50 uncompensated negative charges in vacuum. Okay, if you put it in a cellular environment, in an electrolytic solution, then of course there'll be counter ions congregating and screening it, some of which will be actually condensing on the surface, which basically cancels the charges, but not all, and we'll come to it, and that leads to a somewhat complicated, and that's one example where the environment and the system is tightly connected, right? The environment being the cytoplasm, let's say, or a buffer solution in which you do experiments. Because there's so much charge on the surface, you cannot separate it from the surroundings. Okay, and the histogram shows you the uniqueness of tubulin in terms of the charge. When you screen most of the charge, what you have left is a dipole moment. And on the right panel, you see also the histogram of all proteins. These are all proteins. And tubulin is an outlier with about roughly 2,500 dBi, 2,500 dBi, which is a physical unit of the dipole moment. For comparison, water molecule has about 1.8 dBi, and so it's water is a polar environment. So that also plays a role. You cannot abstract it out because water um, will attach to the hydrophilic surfaces, and most proteins have hydrophilic surfaces, tubulin also, and will create a complex network. That's still uh, largely, I wouldn't say unknown, but not really investigate in great detail what this network looks like. But roughly speaking, it would be, the negative charges on the surface will attract the positive charge on the water molecule, which is the hydrogen, right? And then you'll have the oxygens sticking outside of the surface and forming a hexagonal-like lattice in terms of the orientations. So it's not random. It's not, and from the point of view of electrostatics as well, because these dipoles will create a lattice with a potential for ordered dipole moments on that surface. So we'll come to it because all of it matters in terms of systems response to electric and electromagnetic fields. All the charges, whether they are in the form of an uncompensated charge, what we call in physics a monopole, or dipoles, or quadrupoles, or more complex arrangements, feel, quote unquote, electromagnetic fields, electric fields, and they will respond to them. And the response function is called the susceptibility of the dielectric constant. Okay, so that's kind of the introduction to this to this work and conceptual framework. And because microtubules play such a major role in cell division, intracellular transport, they also modulate ion channels, by the way. Um, let me, okay, we'll come to it. But one specific aspect of the tubulin dimer, oh, here it is. On the left-hand side, you can see the alpha-beta tubulin heterodimer, and you see these C-termini, which are sticking out. C-termini, which are facing the environment, the solution in which microtubules are located, C-termini contain about 40% of all the charges, electric charges, and they are flexible. So you can think about them as antennae. And so they will respond to electric fields and electromagnetic fields. And coming back to this, it says here that microtubules affect ion channel activity, modulate. And C-termina in particular interact with voltage-dependent ion channels. So they can open them or cause closing, etc. The more complicated aspect of these interactions is how electromagnetic fields. So these are oscillating electric and magnetic fields propagating with the speed of light, roughly less in dielectric media, but comparable speeds. Uh, and and then and then we'll talk about this because that opens a Pandora's box of possibilities. Electromagnetic fields span many orders of magnitude in terms of the frequencies. Let's say from low, single digit hertz, low, and then again, if, if we want to talk about effects on the human body, we have to understand the processes in the human body occurring on similar time scales or frequencies. The inverse to the time is frequency if the process is cyclical. So when you look at processes which may couple to external electromagnetic fields, you should typically look for corresponding time scales or frequency scales. And there will be different, very vastly different responses in terms of physiology or cell biology, depending on the frequency. As I mentioned, starting from single digit hertz, which would be like corresponding to the heartbeat, heart rate, and then you have kilohertz, which would correspond to, let's say, uh, enzymes producing ATP, FATPases. So this is kilohertz, milliseconds. And in between, you'll have um, EEGs, which are, the cyclical processes in the human brain on the order of, let's say, from 10 to 40 hertz or so. And then you go up to hundreds of kilohertz, hundreds of kilohertz, megahertz, gigahertz, terahertz, and then visible light, which is 10 to the 15th, 16th, and ultraviolet even higher. And now physics kicks in because, from quantum mechanics, we know, Planck's formula, energy is proportional to frequency through the Planck constant. At high frequencies, you have quantum processes, photons, quanta of light. At low frequencies, you have largely classical, slow processes. Now, so all of this interacts, and we examined some of it in terms of microtubules, how these interactions affect microtubules, and uncovered some interesting behaviors. This is a different view of a microtubule. On the left, you see, and that's from many years ago, a study published in 2001, which was actually groundbreaking at the time, one of the most computationally intensive. That's not an artist's rendition. This is a computer representation of the electrostatic surface of microtubules with atomic resolution. So every atom was represented, and each tubule dimer has about 10,000 atoms. So the microtubule will have millions of atoms, and each one will contribute its electrostatic properties. So you can see here, red is negative charge, blue is positive, and you can see how, in this case, symmetrically, beautifully represented it is. And the two ends, the plus end and the minus end in panel B and C, are not identical. And biologists knew for decades that the microtubules are asymmetrical. The plus end and the minus end are different. The same, by the way, is true of actin filaments. They have the pointed end and the barbed end. And for microtubule, you say the plus end or the growing end, the minus end, the shortening end of a microtubule. But in the upper right hand corner, you can see the surface is a bit more nuanced or more detailed, distributed in terms of the charge. Is much more messy if you really zoom in. And you can see also these nanopores, black spaces in between dimers where actual molecules such as paclitaxel, which is used for chemotherapy, penetrate through and go inside the microtubule into the lumen. A lot of interactions take place on the surface with other proteins, including motor proteins. So this is electrostatically driven to a large degree, as well as between tubulins to form the microtubule. All of it has a large component of electrostatic interactions. And the same authors of the paper later on showed that, for example, for actin polymerization processes, if you were to turn off electrostatics for some, computers could do it, but of course in the lab it's not so easy. Then the polymerization process would be reduced by something like a third, roughly speaking, 30% to 40%. So electrostatic accounts for at least that much in terms of the rate of reactions. It's short range, by the way, because it's screened typically. Okay. And this is just showing you that something I mentioned before, namely polarizability, which is, what does it mean? Polarizability means the response of the system, and now we are abstracting it slightly, to an electrical or electromagnetic field applied externally. And polarizability can cause several distinct effects. One is, as you can see here in the middle, C termini, they flail around, and electromagnetic fields will affect their motion, which could be disruptive for motor transport, for example. The second thing is charges, whether individual charges or in form of dipoles. will respond to the fields, and positive charge will move to the negative field direction and vice versa, right? Positive charges to negative um, will be attracted. So that's kind of dipole stretching, charge movement, and then you have also reorientation of dipoles to align with the electric field. So stretching, reorientation, alignment, and then finally water molecules I mentioned to you, and there's a slight at the bottom right-hand corner you see a water molecule with the charges and 1.8 dBi dipole moment. Water molecules will reorient themselves, but if the field is oscillating, they will oscillate with the field. So that's polarizability, and we actually were lucky enough to collaborate with um, um, Professor Dogariu from University of Central Florida, where we actually measured for the first time very accurately several things, including polarizability um, as a function of pH. So you see, this is the first indication that the medium cannot be separated from the system, because if you change the pH of the medium, you change the polarizability of the system, and this is shown as beta of tubulin. Sorry, and you can see how that changes. Actually, it's even hysteretic, so it depends on the change of pH. You have a different value, so it has a hysteretic behavior. And the same here on in the right-hand panel, you see the dipole moment of tubulin ranging from 2000 to 1920 or something, as you change pH from only 6.5 to 7.5, which is all very physiological, right? I mean, physiologically, pH even can change to down to 1 in the gastrointestinal system and so on. So that, and then the net charge changes with pH. Why is that? Because some of the amino acids can be protonated or deprotonated, so the charge on the system can vary depending on the pH. Okay, so that's the first experimental data showing you that there is system is composed of the environment as well. And these are some illustrations of the microtubule drift with electric fields. I'll show it again. You see how microtubules glide and drift. This is random at 0 field, and then you increase the field to only 50 volts per centimeter, which is not a lot. It's actually comparable to the fields in the cytoplasm, and they move with the field. And there's some micrographs from higher electric field experiments. But there's more, because in the presence of higher electric fields and electric fields with different frequencies, electric fields which are alternating, there are some additional effects seen: realignments, reorientation of microtubules, and even formation of some non-trivial structures. This is now with frequencies, frequencies going up to 2.5 megahertz. We explored even higher frequencies, as you will see later on. So back to my first few minutes of comments. The intensity of the electric field or electromagnetic field is the first parameter that affects the results. The second is the frequency of the electromagnetic field. The third one is orientation with respect to the microtubules and the cell plane in which the cells are formed, or not formed, but present. And the effects involve dielectrophoresis on microtubules, electroosmotic flows, because there is some electrical gradient formation, and then thermal conduction, small but non-zero. So these three are the main physical effects affecting the orientations and the formation of these. So now the next element of complexity is structure formation. So external fields, and now affecting internal structures. And we start from tubulin, which is the building block, into microtubules, and microtubules then form, as anybody who's taken cell biology knows, form various structures, not just microtubules per se, but also centrioles, centrosomes, axonemes, the various um, um, composite structures, composite made of microtubules in various arrangements, the centriole being probably the most intriguing and important because it's touted to be the eye of the cell or the processing unit in the cell. And I will leave it at this just to um, to move on. But so at different frequencies, you will have different mechanisms. Um, and I'm looking at sort of resonant because all frequencies will contribute, but some sensitively and some not so sensitively. Okay, so you have the range here. Okay, and different structures, the rule of thumb in physics is this. The larger the structure, the slower the processes. So the lower the larger the structure, the lower the frequency that interacts with it, right? So small things like atoms will strongly interact with visible or near visible, near infrared molecules, maybe 10 terahertz, water especially, and then the bigger it is, the lower the frequency that you should be looking at for significant interactions. Okay, and so we did the first group of uh, dynamical experiments involved conduction, and we suspected that ions will flow along microtubules, and so microtubules can form conductive biological wires inside the cell. And I think we proved to be right, as I'll show you in a second. But we still don't know whether what's flowing along microtubules is just ions like sodium, potassium, or maybe also protons, or maybe even proton jumping from water molecules, or maybe even electrons through the protein, which goes back to speculations of St. Georgi in the 50s, I believe, that proteins can be conductive electronically. And I believe that may be the case under some specific conditions. Okay, so we did experiments on microtubules and tubulin under various conditions, different ionic strengths and different tubulin concentrations. And that was very eye-opening because we found a lot of things to our surprise. But before I show you the results that surprised us, let me just say that microtubules may also interact with ions in different ways. Number one, they can impede the flow of ions because they're just very bulky obstacles in the otherwise, conductive fluid. And then they can also attract and condense ions, which is known as the Manning theory of electrolytic solutions. And then they can also accelerate or conduct ions along their lengths. So three is condensation, and four, finally, you have C-terminal. I remember are very flexible, and they can cause fluxes and even act as we call batteries. And yeah, there were some experiments. This is kind of a summary of different experiments. The values of conductivity of microtubules in Siemens per meter, as you can see in the literature, varies by 6 orders of magnitude. And I suspect much of it is due to the experimental conditions, which were different. So you see, the system is part of the environment, and if you're not careful, your measurement may not reflect the system per se, but the environment in which the experiment was conducted. We actually, yeah, we'll come to it. We've done experiments. First of all, almost 20 years ago, these were the first surprising results published in Biophysical Journal with Horatio Cantiello, who did the experiments, and we did some computational explanations. It showed microtubules. In the micrograph, you see a single microtubule grabbed by two micropipettes with electrodes, and signals, electric signals, were sent from one of the electrodes to the other, and the intensity was measured, and we found to our surprise that the signal was increased. In other words, a microtubule acted as an amplifier. We call it a transistor-like amplification of ion flows. That was really interesting and intriguing. But there was a single microtubule um, and and a signal or a pulse. And we did some computational predictions. This is from the modeling representing microtubules as cables of these RLC networks. Shown here, a single dimer was represented as a combination of an inductor, two resistors, and a capacitor. Capacitor is shown with an arrow across, which means it's actually variable capacitance. And why is that? Because capacitance is due to the formation of a double layer. The surface of a microtubule has a negative charge, as I told you, roughly. There's some positive, but mainly negative. And then the second layer is counter ions, positive potassium and sodium ions, forming the second layer separated by something called the Debye length, which under physiological conditions, the separation is about 1 nanometer, maybe 9 angstroms or so. And then if you know physics, electricity, magnetism, first year undergraduate, that'll create a cylindrical capacitor. But there's also resistance because it's viscous fluid, so there's resistance along the flow and away from the microtubule. And the inductive part is due to the geometry of microtubules is cylindrical, but actually the arrangements of tubule dimers are helical. So you can create a solenoidal flows, and that's why there is an inductive part. So this is an equivalent electrical circuit, and we predicted these values and waited about not quite 20 years, but maybe 17, 15 years to find some validation of these numbers, which were predicted at that time. And yeah, I'll just skip the models because they lead to some very interesting nonlinear differential equations, which I like a lot, but maybe they're not so important for your audience at the moment. These are the beautiful and complicated differential equations which we solved that describe the voltage pulse propagating along a microtubule in the form of this increasing amplitude solitary wave. Okay, and that was a bit more detailed representation later on. 2 years later, we published a physical review paper on a detailed representation of microtubules in cylindrical geometry with C-termini and so on. Okay, this is from you, Mike, and I show it a lot because it's a very powerful diagram showing how transmembrane potential changes depending on the cell type and the normal versus cancer cells will have different, and this is one of the hallmarks of cancer, by the way. We published a review paper on electrical properties of cancer cells, and that's one of the electrical hallmarks of cancer cells. Another one would be, lower pH outside the cell and higher inside, and some other things. And these are the charge properties of cells, or constituents of cells. Inside, you have the potassium, sodium, chlorine, calcium, magnesium ions playing the main roles, and then proteins that are, as I mentioned, most electrostatically important are tubulin and actin, and so on. So all of this forms, and don't forget water, which is dipolar. All of it forms a very complex electrically and electromagnetically structure, which is dynamic in response to external fields. And we know about voltage-dependent ion channels. You work on this, a lot and contributed tremendously to the field. But what happens inside is still, to a large degree, a mystery. Okay, and so in the next few minutes, I'll try to cover what we discovered in terms of microtubules, and here are the micrographs of our experiments with 3 different concentrations of tubulin. You can see here microtubules labeled fluorescently, and they show up as white streaks. It looks a bit like spaghetti, I guess, or some random networks. But
This is in vitro, right? This is a cell-free system here.
yes, this is in vitro, exactly, purified tubulins. Um, and you have the scale. But what's important to note, and I'll come back to it in a second, is the numbers of microtubules, as you can see in the samples, increase by a factor of 10 from the left to the right, and then 100 again. So another 10 to the extreme right. So you increase the numbers of these conductive, capacitive, and inductive units from the electrical point of view, and you would expect something to scale with this, right? You would expect corresponding or roughly corresponding changes in conductivity. And we did these experiments. This is kind of a schematic of the experiment with the semiconductor characterization device called Keithley. And we found, okay, we found, first of all, frequency dependent responses, conductivity. And by the way, we compared tubulin to microtubules. In other words, polymerized, unpolymerized protein to polymerized, and there was a big difference, the same amount. But forming polymers increases conductance here. Green is microtubules. And also with the number of microtubules that increases, but maybe not proportionally. And then there is also the aspect of capacitance, which we were able to measure as a function of frequency later on in this experiment that was published not so long ago, 5 years ago, 2020, in these experiments. That's the setup. And these are the results. So blue, you have microtubules, and red is tubulin, unpolymerized protein. And you can see the dramatic difference. So with the capacitance, which is imaginary part of impedance, speaking the language of physics, is higher for microtubules than free tubulin. And then resistance in this experiment is greater for microtubules than for tubulin. So they are more resistive. But this is at, and I should tell you, we did 2 sets of experiments with opposite results. At low ionic concentration, which was 20 times lower, not physiological, something like 4 millimolar, so let's say sodium, you have microtubules being highly conductive but not highly capacitive. At physiological or close to 80 millimolar, the opposite happens. They are more resistive and more capacitive. So in other words, microtubules change from, at low ionic concentration, from being very efficient conductive wires of ions at low ionic concentration, and I'll explain in a minute why. At high ionic concentration, they trap ions and become capacitors. They store charges, counter ions. So you see, from dynamic movement, so conductivity is basically movement of charges. Capacitance is trapping of charges. So high concentration of ions, they trap them. Low concentration of ions, they conduct them. And the question is, at what point does it change? What's the crossover point? I'll just cut to the chase because the time is running out. But it's very important to keep this in mind, because the cell may use it controllably to use microtubules for either conduction or trapping of ions or charged objects. Okay, and okay, these are some additional experiments, and before I explain to you why this is so in a minute, I just want to come back to the fact that when you see this on the left-hand side, you see the concentration of tubulin being increased from 0.22 to micromolar to 10 times that to 100 times that. Yet the capacitance of the system is virtually the same within a few percentage points. 10 to the minus 5, 10 to the minus 5, 10 farads for the system. And then you have errors and so on, small errors actually. And then resistance is also roughly the same because 9.74 10 to the fourth is approximately 1 times 10 to the fifth. So you see something interesting, still unexplained happens, that in spite of all of this, the networks themselves have some sort of stability of resistance and capacitance in spite of increasing the numbers of microtubules by large numbers. But we were able to extrapolate these values to the 20 micrometer microtubule at the bottom in red, and they correspond quite favorably to our predictions from 15 years prior. Okay, so I'll just skip it. But let me just, in the interest of time, as I'll try to finish in a few minutes, show you how microtubules move in response to the changing polarity of the external fields. And also tubulin, by the way. And then we also measured electrophoretic mobility of tubulin, so mobility with respect to electric field, an electric field applied as a function of pH. And there's something very interesting happening here too. Namely, at pH 5 or so, microtubules become neutral. And below, they actually turn to be positively charged. And I kept telling you and your audience that they are negatively charged, but at pH 7, yes. If you lower pH, they become neutral and then positively charged. So that's another dial you can turn. And if you want to do experiments, change pH and properties change as well. Change the ionic concentration, if you remember, they also change from conductive to capacitive at high ionic concentrations. Okay, and that's what zeta potential shows. And okay, let me just try to explain the change, and that'll probably be the end of my talk today. So there is something called the Poisson-Boltzmann equation that if anybody in your audience is interested in how to model these things, this is basically an equation that solves for the electric potential around charged surfaces surrounded by ionic solutions, because ions respond to the electric field of the fixed charges, in this case fixed on the protein surface. So we solved this for the cylindrical geometry of microtubules and the dielectric constant of the medium, and we found that from that you can calculate the fraction of the bound ions to the surface and how their concentration changes as you go into solution. And everything depends on the bulk concentration of the salt in which the microtubules are bathed. So you see here the lumen, which is inside, how it depends on the distance to the surface, outer surface of microtubules, and C-termini. And this is kind of a summary graph. If anybody wants to get into more detail and read the paper we published in Frontiers in physiology, I believe it is, and it shows you how everything depends on the bulk concentration and the surface from which you measure. And this is the bottom line. From that you can calculate the conductivity, and you can see here in the right hand side panel, the red straight line is the conductivity of the solution itself as a function of concentration of potassium ions. In green, you have microtubules. Conductivity of microtubules, or around microtubules, I should say, as a function of bulk concentration. So at low concentration, they are much higher conductive than the medium. You can see here it's actually 100 times or more, a few hundred times more conductive. But then there's a crossover point at about 100 millimolar, which is close to physiological. between 70 and 120, depends on the conditions. So this is a tipping point where microtubules become either more conductive or less conductive than the cytoplasm. And that's interesting because a cell can use it for whatever function. And the explanation of it has to do with what's called, I mentioned the Debye length. The Debye length is the distance from a charged surface um, over which electrostatic dominates thermal fluctuations. Beyond the Debye length, basically the ionic solution doesn't feel the surface charges of, let's say, microtubules. And that value depends on the bulk concentration. And in the case of microtubules, as I mentioned in the beginning, everybody says, oh yeah, the Debye length is 9 angstroms. Yes, at physiological conditions. But if you lower the ionic concentration to 0, that's infinity. When you lower it to 4 millimolar, it's about 100 nanometers. This is a number which fluctuates. Not fluctuates, but changes depending on the conditions. Again, the message of today, the environment is part of the system, and the system responds according to in which environment it's placed. So maybe I'll stop here and I'll let you ask questions. There's some more results, but I think I've given you probably quite a lot to talk about.
Yeah, remarkable. So one thing that I'm very interested in is let's assume for which I think seems very likely given what you've shown, that the microtubules implement an electrical network in addition to whatever biomechanical network they have. So what we have then is an electrical computational network that is also continuously sort of refactoring itself in terms of structure. So this is, a very different architecture than any kind of computational system that we make now. Much more, well, for obvious reasons, much more relevant to the biology, which is also very, very plastic in terms of, for example, the bioelectricity that we study. Cells are moving around all the time and so on. So how much work has been done so far in asking the question, like what are the like if you make a somewhat of a biorealistic model of these things, what are the computational capacities of, I mean, I know you've done models of memory and things like that, but
Yes.
but what are the computational properties of such networks? And as they do start to move around, I mean, I think the cytoskeleton is a really good candidate for things like a memory mechanism, for example. But the missing piece is to show, despite all the things that a cell is constantly doing to rearrange and move around, refactor that cytoskeleton, how does information stay solid? And I don't think it needs to stay unchanged, because I don't think that's what the biology is doing. I don't think it's locking down information with error correction codes and things like that. I actually think it's doing something quite different about it in terms of interpreting it on the fly. But nevertheless, there needs to be some computational modeling of what's actually possible with things like that. How much of that has been done?
Yes. Well, a little bit. And it's you're absolutely spot on in terms of understanding this as a dynamical computational system, as opposed to, let's say, silicon-based computers. It is a system that can be evolvable, erasable, reprogrammable. The architectures can be flexible and controllably rearranged. And I'm sure the cells are doing that. We know because dividing cells need microtubules mainly for point-to-point transport and cell division, mitotic spindles, in which case they are oriented very precisely to the chromosomes, and there is, I would say almost certainly there's electrical signals or pulses sent maybe for synchronization of segregation of chromosomes along that will make a lot of sense. Now, in neurons, it's a different story because they are stabilized, they form parallel bundles, and these bundles can perform additional computational um, um, operations in addition to action potentials, everything we know about neuronal computing. I think I call it a computer within a computer. That is, microtubules in neurons compute something, and maybe they change the response function of the neuron to neurotransmitter release and things like that. Now, How much? In one of the papers, we did estimates of how much information you can encode in a microtubule, and that varies depending on how many bits per tubulin, and it could be a lot. But the simplest version, which integrates all of this, what I said, namely electrical encoding and decoding, would be C termini, in at least two states, straight out or bent.
Mm.
They can actually, we simulated that. And if you assume that, then a neuron would have something like a gigabyte of memory through microtubules alone. Compare it to a one bit switch off and on in classical neurophysiology. Now we go 9 orders of magnitude higher. Each neuron can have at least 1 gigabyte of memory through microtubules. And since there is what is it, 100 billion neurons, roughly speaking, in the human brain, then if that was the case, you could encode 10 to the 20th bits or bits of information through microtubules. And again, that's 9 orders of magnitude greater than just taking neurons as switches, which I think is an affront to the neuron, to be quite honest. Yeah, so that's the simplest, and just assuming something we know for a fact. But there could be additional degrees of freedom. We wrote another paper with Travis Craddock and Stuart Hameroff on calmodulin kinase too, which, so another level of encoding could be through phosphorylation, which is well-known signaling in biology, right? And so these enzymes phosphorylate tubulins, and then you could have actually even more, vastly more, at least 1,000 times more information bits if you take this into account through microtubules, and we know that they are phosphorylated by this enzyme CAMK2. So what I want to say is there could be more than one channel of information encoding, decoding, and information processing. One could be slow, maybe enzymatic is slow. Another could be fast, electrical, and another could be electromagnetic, which is super fast. And that's the second part of this talk, which I didn't have the time to talk about because the time is limited. Um, but um, but it exists, and we've done some experiments with Greg Scholes at Princeton and Arat Kalra, my former student, on looking at, let's say, specific information, quantum information storage in microtubules by using tryptophan residues, of which there's 8 per tubulin. They can be excited by electromagnetic waves. So that would be yet another level of fast information processing and quantum state formation. So yeah, it's a huge, huge number of possibilities.
Yeah, amazing. Amazing. Well, I think there's a really great opportunity here. Um, to use these biorealistic models to then either make them a reservoir in the sense of reservoir computing, or try some kind of more traditional connectionist architecture on them, that kind of thing.
Yeah, and so these are glimpses. We don't have a fully blown,
Yeah,
theory of this, even a model. But each glimpse gives you, in my opinion, another possibility of using it for various applications.
that's
And
right.
I'm sure the cells over the past 2 billion years or so discovered how to do it.
Yeah, yeah,
We're just trying to second guess the cells.
yeah. Yeah, amazing. And that's before putting in all the synbio stuff, right? About changing the actual, tubulins working in. I mean, there's a whole other dimension here.
Oh, yes, I forgot to mention, and so obviously this the cells have a repertoire of tubulins. There's
Yeah,
20 genes encoding for tubulin in the human cell. And of course, they can express different isoforms of tubulin differently. And each isoform, by the way, has a slightly different electrical property. And most importantly, each isoform of tubulin has a different C terminus with different numbers of charges and numbers of residues. So yeah, the number of possibilities is astronomical. And by the way, that can be technologically exploited if we were able to. Well, we can express tubulin recombinantly and make it into these computer architectures based on proteins.
Yeah, yeah, amazing. Super interesting. Yeah, thanks so much. That's fantastic. Really an amazing. I mean, you've opened just that whole aspect of biophysics is I think is going to have a huge potential into the future.
I hope so, and I also want to mention that this is not in isolation because what you do with ion channels and membranes is a layer which interacts with this internal layer, and especially in neurons, of course, that's obvious, right, with action potentials, but not only in neurons, because if you modulate ion channels and change the ionic concentrations inside the cell,
Yeah.
that's the environment that microtubules live in. So you
Mhm.
see, it's a two-way communication.
Yeah. Yeah.
And
Yeah.
and that is the biophysics of the future. I think we can integrate the cellular, the cytoskeleton aspect with the membrane and ion channels into one interacting network.